Hello everyone, my name is Nicole and I'm part of the Biomedical Sciences research group at UHI. And today I'm excited to introduce to you some of the tools that we use as part of our everyday research. So within the Biomedical Sciences research group, our research spans across many different disciplines, from the intricacies of genetic disorders of cancers to broader environmental and metabolic studies. We're based on the Inverness campus within the new Life Sciences Innovation Centre building and Anne Lockram. So the first tool that I'm going to discuss with you is cell culture. And this is a really commonly used tool in research, and it forms the foundation of many experimental procedures. This is where we culture cells within a controlled environment, and by using a variety of cell types and conditions, we can conduct really extensive research into looking at disease mechanisms and potential treatments. You may have heard of HeLa cells, and these are some of the oldest and most widely used human cell lines, and they're derived from the cervical cells of Henrietta Lacks. This was done back in 1951, so these were the first immortal human cell lines ever grown in culture. This means that they can divide indefinitely, and it makes them a really invaluable tool for scientific research. HeLa cells have been really instrumental in many scientific breakthroughs. They helped the development of the polio vaccine, and they've really helped progress cancer research and generally just progress our understanding of cell biology. They're named after Henrietta Lacks, and there's been a lot of media about these cells in the past, and there's even books and movies which go into the scientific breakthroughs of these cells and some really important ethical considerations. And I'd advise, if you're interested, to go and check these out. From HeLa cells, cell culture has advanced dramatically over the years. And we're now at the point where we can culture organoids. Organoids are three-dimensional, miniature, simplified versions of organs, which are cultured in vitro, meaning outside, so in the laboratory setting, and they're derived from stem cells, from, for example, skin cells which have been reprogrammed. And they're really, really useful, as you can imagine, to be able to model organoids in a laboratory setting. At UHI, we culture a variety of different cell types, from stem cells derived from skin cells, different types of immune cells such as B cells and macrophages, skin cells themselves, cancer cells, so cells extracted from tumours, and we have these various types of cells which are from either healthy patients or cells which model a specific disease. And these really form the starting point of many of our experiments at UHI. The second tool I'm going to discuss is spectroscopy. And this is another vital tool in our laboratory, and it allows us to analyse molecular composition of materials and quantify biological substances with a very high precision. There's many types, such as Raman, which is used to measure vibrations, and colorimetric assays, used to measure the depth of colour. So at UHI, we do both Raman spectroscopy and colorimetric spectroscopy. So for example, I routinely use the Bradford assay, which is a type of protein assay used to determine the protein concentration within a sample of interest. So I have my sample of interest. I then apply a Bradford reagent, and a chemical reaction will take place. So there will be a colour change within the sample, and the colour change will reflect the concentration of protein within that sample. So this is a really helpful tool, and it gives us information about, I've given the example of a protein, but there's other things such as cell toxicity, which you can measure and give an indication of what is in your sample. Antibody tests are commonly used in laboratory research and in a diagnostic setting. So they're really crucial, and they help to detect the presence of specific antibodies. And these are proteins which are produced by the immune system in response to an infection. So you may remember the COVID-19 tests. These were able to detect antibodies which have been produced against the SARS-CoV-2 virus. So they help to determine if an individual has been exposed to the COVID virus. There's similar types of antibody tests which are commonly used in practice, such as the HIV antibody tests. These are able to identify if there are antibodies against the human immunodeficiency virus. There's a malaria test. These are able to detect if antibodies are present in the sample that have been produced against malarial parasites. ELISAs are a type of antibody test commonly used in a laboratory setting. These are enzyme-linked immunoabsorbent assays. In summary, they are antibody tests which take place on a culture plate. You can see one on screen, and that's a plate there with 96 wells, and it helps to ensure that the antibody tests are taking place in a very controlled environment, and they give quite crucial and accurate results. At UHI, we do many ELISA assays, and we publish multiple publications using this tool. Our most recent, this was published by Dr. Ryan McLean this year, and it was able to show that schizophrenia patients have an increased level of antibodies against a certain receptor in their sample. And this is helpful because it helps to show that these schizophrenia patients have a particular biomarker, and this could potentially be used to diagnose schizophrenia in the future. Mass spectrometry is another common tool which we use at UHI. This is an analytical technique used to identify and quantify the composition of the sample by measuring the mass to charge ratio of ions. Really, this is just a fancy weighing scale, and it helps to provide precise measurements of molecules, and it's really crucial for various scientific applications. For example, it's used in space missions, and it helps to analyze the composition of materials found in other planets. It's used in medical diagnostics, so it helps to identify and quantify biomarkers related to a disease of interest. It's really useful in forensic science. It helps to identify substances such as drugs and toxins which have been found on a crime scene. And quite famously, it was used back in the Manhattan Project, and this was used to separate isotopes of uranium, and this was a critical step in the development of the atomic bomb. At UHI, we simply use mass spectrometry to help measure molecules within a sample, such as lipids and proteins, with very high precision. And it helps to understand biological processes and disease mechanisms. So, for example, I published papers where I've measured proteins from immune cells following activation. And so I help to identify what proteins have been increased or decreased following immune activation. PCR, otherwise known as polymerase chain reaction, was a really revolutionary technique in molecular biology, and this helps to amplify specific DNA sequences. And so it helps scientists to generate millions of copies of a particular DNA segment, and it's used in many applications. For example, PCR is used in genetic testing. It helps to identify genetic disorders and mutations of interest. It's also used to analyze ancient samples. So it can amplify degraded DNA from ancient specimens, providing insights into evolutionary biology. It's often used in organ transplantation to help match donors and recipients. And it's helpful to diagnose various diseases, such as cancers and infections. In forensic science, PCR helps with DNA typing. So it helps to match individuals' DNA with a crime scene. At UHI, we use PCR to measure the gene expression of various genes of interest in various samples. And this helps to understand how gene expression may change in various conditions and diseases. And it helps to therefore understand disease mechanisms and identify potential therapies. Histochemistry. This bridges between biochemistry and histology. It helps to examine tissue architecture in very fine detail. And it's really important in diagnosing complex diseases and understanding disease pathologies. So whilst histology is the study of microscopic anatomy or biological tissues, therefore this helps to look at tissue architecture and cellular structures and give insights into pathological changes which are associated with disease. Combining this with biochemistry, we can use staining, such as various dyes and antibodies, to detect certain proteins or lipids and molecules of interest. And histochemistry can give insights into various aspects of a tissue or cells. So it can help to look at the organization of different cell types within tissue and how structural arrangements may be changed between a diseased tissue or healthy tissue. It can also give information on individual cells, such as their shape and their size and their internal components. It can detect certain proteins of interest, which may be associated with a disease, also carbohydrates and lipids of interest. And this is commonly used in diagnosis. So you can use this technique to understand and identify various subtypes of a disease, such as subtypes of cancer. You can use this to monitor skeletal muscle abnormalities. At UHI, we use this technique in cancer research. So we have tissue specimens from control tissue and tumor tissue, and we're able to use this technique to monitor changes within the tissue between the healthy and the tumor tissue, and really give insights into the pathology and what's going on within that cancer tissue. So thank you for your attention. I hope you enjoyed learning about some of the tools which we use in the Division of Biomedical Sciences at UHI. Please feel free to go on our website and find out a little bit more about us, and there you will also find our contact details. So please feel free to get in touch if you have any questions. Thank you.